I'm going to continue more or less where we left it off yesterday. And the plan is, I did a lot of talking yesterday, but today I'd really like to calculate something with you. And just like Rob was emphasizing numbers, as in raw numbers, I'm going to emphasize functions and try to understand how things that we are interested in depend on the relevant parameters. Okay, so, but yesterday we did go over some numbers with an implicit understanding that these numbers have gigantic error bars for mutation rates, for distribution of phenotypic effect sizes, et cetera, et cetera. But what we didn't talk about was natural selection itself. And we talked about fitness and how non-trivial it is as a concept and a quantity. And we've gone through various caveats. But now we're just going to take the most naive point of view of it. And so the simplest view of fitness is the expected rate of growth of the population and see where it will take us. It is a sensible description, at least over the short spans of time where environment is reasonably stable. So let's talk about dynamics of natural selection. And my first question is whether you can see any of my scribbles. And you can. You have eyes of an eagle. Terrific. So, okay. There is some distribution of fitness in the population, right? That's the case that we're interested in. Yesterday, again, discussed at length that we expect in any interesting cases that there'll be many genetic variants in the population running at the same time, and these variants are not non-neutral, so they affect growth. So therefore, if we call this rate of growth x, then there'll be some distribution. There'll be some number of individuals falling into a bin of fitness dx, right? And then our question is, what is the dynamics of this distribution? And well, we just defined fitness as the rate of growth. So there we have this exponential growth in each bin. But obviously, that can't be the whole story. This way, the whole population would be exploding in size. So there has to be something limiting the growth. And that depends on, presumably, the whole distribution. And I'll write it like this. So there's going to be some density-dependent death rate defined in such a way, or rather trying to parameterize the fact that the environment has a finite carrying capacity, and at very large total numbers, the population is going to start dying off. And the simplest model assumption is basically to say that total population size is actually constant. So what we want to assume is that the total number of individuals in the population is constant. And a very simple way of achieving that is to replace this density-dependent death factor by simply the population average of the fitness, so defined as the integral of a distribution, like that. So now what we have is a population where individuals that are fitter than average are increasing in number, and individuals that are less fit than average are decreasing in number, and the whole lump of a distribution is shifting towards higher fitness. So so this is actually sort of both intuitively appealing, but rather non-trivial statement here, that the fitness is a relative notion. So, think of this old joke about two campers. They wake up in the morning, they see the bear coming. One of them rushes to unpack his sneakers, put the sneakers on, and the other one says, "Why are you putting on the sneakers? You know you can't outrun the bear." And he says, "Well, I only need to outrun you." So that's the thing. So here we have the selection of the fitter. It is the relative notion. So and you can happily check that with this definition now, if you just integrate our simple equation that I'm going to rewrite, if you now integrate it over x, the right-hand side will average out to 0, and the total population size is conserved. So that's a very simple manipulation. But let's do another simple manipulation. And that simple manipulation goes by the name Fisher's fundamental theorem of natural selection. And Fisher's fundamental theorem of natural selection addresses the rate of evolution here. Rate of evolution defined as the rate of increase of that mean fitness, which evidently here is shifting to the right. So let's call this, this is sort of the velocity of our traveling lump. And let's calculate it. Well, to calculate it, all we need is take this equation, multiply it by x, and integrate, right? So evidently, this is the mean rate the rate of growth of mean fitness. And on the right-hand side, what we have is dx, x squared, and x. And the second term, you easily convince yourself, is just going to be the average squared. And that thing will reassemble as x minus the mean, right? Relative fitness squared averaged. So that's the variance in the distribution, right? The width of this distribution, it sort of embodies Darwin's insight that natural selection requires standing variation in the population. It's the celebrated result, and that's basically all there is to it. It took us 10 minutes from start to finish. But we aren't even finished yet, because we can make a little stronger statement. That's a pretty simple equation to solve. And we can find a class of solutions to it. We can basically look for steady state solutions which correspond to these translating lumps, or let me call them Fisher traveling waves, or Fisher traveling waves. Traveling wave. I'm not sure that calling it Fisher traveling wave is actually standard terminology, but I like it that way. So this is a little bit of homework. You can convince yourself just by inspection that this little equation has a Gaussian as a solution. So in here we of course have the time-dependent mean, which we've already determined, right? It just increases linearly with time at basically this rate, right? So that's a perfectly exact solution, and not only it's a solution, it's actually an infinite family of solutions, because Fisher's theorem relates the rate of adaptation to the variance, but it doesn't tell us what the variance is. So we have, so basically any Gaussian lump in this approximation will just happily evolve at the constant rate. So the only thing is we actually have to figure out how fast it's going and what the sigma squared is, and that's the whole problem. So, okay so far? You with me? Okay. So let me erase something. And let's start thinking about this question of what the variance is. And of course, the first thing that comes to mind is, okay, let me erase a little more. And let me erase a little more. No, I'm not going to erase more. I'm going to make another little picture of this. So we got the Gaussian solution for a Fisher wave, and it goes as and e to the, okay, let me call this delta x. I'll just define x relative to the mean, squared over to sigma squared, something there. And we observe the following thing, that for any finite n, in any finite population, if we go too far into the nose here, so in fact, let's just do this. Let's pick some value of x and ask, how many individuals are there with fitness higher than that value? And clearly, if we push this far enough, for any finite n, that number eventually is going to be less than 1. Okay? We can't have a fractional individual. So, therefore, this distribution has an edge. There is a maximal fitness in any realistic distribution, and we can actually very quickly estimate it, right? All we need to do is, well, do the calculation that I just said. Let's integrate this thing from x max to infinity and require that it's equal to 1. Another little homework exercise, and you will convince yourself that this maximum fitness depends logarithmically on the size of the population. So, okay, wait, so square factor of 2, right? Basically, to see where the logarithm is coming from, right, you basically say that the numerator here times n is of order 1, and right, that will get you the log, and will get you, of course, the scale for this maximal fitness, which is set by the variance itself. Okay? So, as we go on, you'll see logarithms every so often. In fact, yesterday, when I was just summarizing results for you, we talked about the crossover from linear dependence on population size to logarithmic dependence on population size. So ultimately, this is all where it's coming from. So, okay, we just discovered that there is a nose to our fitness distribution. Yeah, please.
How should we understand theoretically that maximum fitness depends on the size of the population?
Excuse me?
How can we interpret that maximum fitness being dependent on the size of population?
Well, you know, just think of a sample. All that's happening here, you're sampling a distribution, right? There is some distribution with a tail. And the deeper you sample it, the more likely you're going to find a rare event corresponding to this sort of very large fitness deviation, if you like. Right? So another way of thinking about this is, sorry, let me just maybe give another, maybe a little bit more biological rather than mathematical answer. So you're looking at individuals, and they have their sort of evolutionary trajectories in the sense that they're accumulating their mutation. And some of them are getting better mutations than others. And we can sort of discuss the validity of this additive approximation that talked about yesterday, but to some extent, whether it's just additive linear accumulation of benefits from multiple mutations, or there is some diminishing return situation that if you're already very fit, it's very much less likely that you'll become fitter still, but you still expect this to be monotonic, right? And this is a probabilistic process, and so the highest outlier that you're going to find ultimately will depend on how many examples you're looking at. You had a question.
This rate of evolution, when you say, do you mean to say that it's an rate of mutation?
I'm sorry. I have we have another question that I already. Oh, you're worried about this. Oh, okay. Okay, I'm doing sort of a little bit of street fighting mathematics like Rob here. So I haven't really solved this this equation for you, right? I just sort of rephrased this relation. So you can right, you can solve this thing on the computer, but better still, you'll sort of realize that this thing is actually sitting in a logarithm, and it doesn't amount to a hill of beans. So you can just iterate this equation, and this will introduce sort of more logarithms for you, right? So for example, I can just write it right here as log n sigma, and then there'll be sort of additional logs, but but eventually we'll just decide that by the time you take many logs, the number approaches 1. There was a question.
Do you mean to say that traveling wave also represent the rate of mutation?
We haven't started thinking about mutations yet. So bear with me for a second. So that's key.
Do you mean actually the tail xm, or do you mean xm minus x bar in that formula there?
I stand corrected. Thank you. Yeah. So where were we? Yeah. So we just said that we have a distribution with an edge, and now we think about what this dynamics is going to do for us, and right, the stuff that's fitter than average is going to go up. The stuff below is going to go down. And but this equation doesn't move anybody right or left, right? It's just a number that's going up or down. So the edge actually cannot change. So this whole traveling lump is going to then collapse into the edge. So if it's to be properly normalized, I know you guys are careful, more careful than me. You're going to correct me, right? So, right, this is the survival of the fittest, in a sense that the fittest is going to fixate, take over the whole population. Game is over. We got no steady state. But then, of course, right, and the variance clearly is going down. Right? Natural selection is eliminating variance. Right? It is picking the fittest. It's killing the variance. Right? So we need some processes that will re-inject the variance for us. And somebody already asked about mutations. So obviously, we have to introduce mutations, and our equation doesn't have mutations yet. So what happens if we turn on mutations and we say, well, we're going to have some process that's going to now move things from bin to bin, and also from the leading edge forward. So now, thanks to this process, some individuals with lower fitness will contribute to the higher fitness, right? So if my S, the mutation effect, is positive, then lower fitness will contribute to the higher fitness bin. So the question is, how much of this stuff is going on? So, well, it depends on the rate of mutation with effect size, effect on fitness S. And we have to integrate on it, right? But of course, the process which is promoting individuals from one fitness class to the next class is going to reduce the number of individuals in a class that it's hitting. So we have this. Okay? So now we introduce mutations. Thank you. Very good. I love this. I have an awake audience. Okay, that's not the only source of variance. Yesterday we were talking about reshuffling genetic variants, genetic polymorphisms between individuals thanks to sex and recombination. So I'll just come back to you in a second, if you don't mind. Okay, so recombination, right? Sex requires two partners. So it's going to sample two individuals from this distribution. So let's call it X1 and X2. And it's going to recombine their genomes and with some probability make a genome with fitness X. Okay? And now we have to integrate over all possible parents and sample them according from this distribution, right? So we're integrating over the fitness of the father and the fitness of the mother with proper weights. And in this approximation, we don't worry that perhaps there is some bias going on, and somehow the birds with bushier tails are preferentially picking each other. Okay, so this is pretty much the whole story, but since I'm writing equations on the blackboard, I may as well try to make them right. So to sort of conserve probability, I have to add another process here, and X and X2. So that just takes right into account so the redistribution of probability so that things are normalized correctly. Okay, so that is recombination. Now I'll take your question. This is before we introduced mutation? Well, so before we introduced mutation, all that was happening, we were making clones, right? We had an individual with a given genome, and all that was happening was it was replicating, and the rate of replication, right? Maybe this would help. So to construct this distribution, we already made a non-trivial step. So if we started completely from the beginning, we would have said that there is a certain number of individuals with a given genome, right? And that number is growing or shrinking depending on what the fitness of that genome is, right? But neither the genome nor its fitness is changing in this process, right? So then we basically just projected this distribution, right? Yesterday we were talking about the space of genotypes being a gigantic hypercube, right? And my distribution here, if you like, lives on this ginormous space of genotypes, right? But we don't like solving equations in ginormous spaces. So what we do is we project the space onto one relevant direction. What is the relevant direction here, right? What's the relevant variable? Well, that's the one that governs the dynamics. We care about fitness, so we squish this whole distribution, we project this distribution onto an axis, and that's what we get, right? Hopefully I answered your question. I think that's great. Well, my question generates, one answer generates two questions. I don't know what's happening, yeah.
Your traveling wave solution, you had mentioned that your x bar is sigma squared t.
Yeah.
And that you said, so it was a solution which was moving without change of variance, but with simply a change of the mean. It was a traveling wave solution.
Yeah.
So that, so that's just, the mean is just shifting with respect to time like that.
Correct.
And in that, that's not this structure, right?
Well, so I guess what I'm trying to tell you is that the solution that I produced for you, this nice, pretty Gaussian solution, is only valid in the limit of n going to infinity. So as long as, if you like, this edge is so far away that you don't have to worry about it, this thing is going to translate, right? So the question is, how far away can it be? You know, if it was 1,000 sigmas away, there'd be a nice, happy stretch of time where we didn't have to worry about this edge, right? The only problem is, in order for it to be, something's funny here, sigma somewhere. In order for it to be very far away, right, the logarithm of n has to be a very large number. N may be 10 to the 10, but its logarithm is only 10. So, there was another question somewhere.
That was not an exact solution. That was only a solution.
Absolutely, absolutely.
Fitness, maximum fitness, it's a function of environment. Is it a function of environment? The fitness,
Fitness is a function of
surroundings,
environment. Absolutely. Absolutely. And this whole story, right? So we're talking about these steady state adaptation, but obviously the steady state does not continue for infinity, right? Again, something else is eventually going to happen. So this only makes sense on the time scale of constant environment, right? So there is an experiment on, I think, selecting for larger ears of corn that has been running for, is it 50 years or 100 years? It's been running for a very long time. And one is actually observing a linear increase in size. Slow, but steady, right? Will probably not continue ad infinitum, right? But in that particular experiment, right, it's artificial, and effort is made to keep the environment constant. But yeah.
So the notion of fitness here has to do with largely the reproduction rate, the increase in population size. But the mutation or even the individual has longevity or stays in that environment for a longer period, will still the results hold?
Yeah, yeah. Well, I mean, yeah, that won't change very much. Okay. So this was pretty simple, but to really make progress, we have to try to solve a little bit more complex situation. And that actually is not the whole story, because this equation is completely deterministic, even though we're describing the distribution in the population. It is still a deterministic equation, and it's okay as long as we have lots and lots of individuals. And each and every bin of this population has many, many individuals. But near the leading edge, right, we define the leading edge by the condition that there's basically a single individual there, or very few individuals, right? So, especially where it matters, we have to be prepared to deal with the fluctuations. So let's introduce the fluctuations into the picture. Yes?
Is there any reason to assume a unique fitness function? Why can't there
Excuse
be
me?
multiple?
Assume
Is
what?
there any reason to assume a unique fitness function? A single unit function, fitness function. Why can't there be multiple fitness function at a time? Why
I'm
can't
sorry,
we assume-
I can't quite hear, even though you have a microphone.
Is there any reason to assume a unique fitness function? Only one fitness function.
Only one fitness function.
Yeah.
Well, to the extent that we define our fitness as expected number of surviving offspring, right? It's just one number. Now, if you decide that environment is changing and this number changes depending on the environment, you will need to consider this number as a function of the environment.
So here we are assuming that epigenetic effect is constant. Is it so? Yeah. Here.
Yeah, so epigenetic effect. Listen, you cannot fit all the biology into this simple equation. And frankly, you shouldn't even try, right? So think of this as an abstraction, right? And think of this as something that defines the behavior of the population on average, right? So, right, we have lots of individuals in that bin, right? With the same fitness, right? We're already allowing the possibility that different genotypes have the same fitness. So you're welcome to consider, and that under some circumstances it will be very important and necessary, that a given population, even if it is genetically identical, has different growth rates. For example, persisters, right? There will be bacterial persisters genetically identical to the growing kind, but they decide to replicate more slowly, right? And at some point, you will want to go beyond this sort of simple collapse of everything into just one fitness and sort of differentiate, so allow the possibility that there are different states. And it will matter if you change the environment again, right? As long as the environment is the same, you can express everything in terms of that one number, right? You change the environment, and suddenly different genotypes will respond differently, and depending on the epigenetic state, even the same genotype can respond differently? Good question. Okay, I got to move on a little bit because otherwise we will not get to even talk about stochastic things. So I'm going to erase and start talking about stochastic effects. So in population genetics trade, stochastic effects are called genetic drift, or, well, let's call it stochastic drift. Stochastic drift. So the easiest way of introducing this, well, actually maybe it's not the easiest, but the historical way of introducing this is in the context of the Wright-Fisher model. And the Wright-Fisher model basically considers a population with some genetic diversity, just for simplicity, let's only consider two genotypes. So genotype 1, genotype 2, genotype 1, genotype 1, genotype 2, genotype 2. And we're going to keep the population size finite. We want to think about larger populations than 6, but I will not go that way. And what we want to know is if we have some probability distribution on, so probability of finding n copies of genotype 1, n copies of genotype 2, I'm sorry, n of G1, okay, right? At time t, what is the probability of finding n prime copies of each of these genotypes? And I'm not going to write too many of them. I'll just put curly brackets to denote that now we're looking at so the probability of finding a certain distribution, right, at time t1 later. Okay. So Wright-Fisher model basically says that if you have a population with this, so think of this population as an urn with some black balls and some white balls. And first let's think about the neutral limit, so that even though genotypes are different, there is no fitness differential. They're all equivalent. So neutral case, right? So in the neutral case, we basically say that to generate a new population, we reach into the bag of an old population and pull out whoever we happen to catch, right? And then we repeat this process with replacement, right? So there is some chance that this genotype is going to be pulled in twice, and I guess this one will go forward, and this one will go forward, that one will go forward. So the probability of getting these black balls pulled out, of course, is proportional to the fraction that they constituted in the original population. So let's basically do this. Let's calculate the probability of going from number n1 of black things and number n2 of white things to n prime 1 and n prime 2. And the probability of pulling out 1 is proportional to the frequency, right? The fraction n1 over population size in the original population, right? So this is our n of g distribution, and here we're constructing the n prime of g distribution, right? And the right. So we're doing this with replacement. So the probability of pulling out n prime of them is just this frequency to the n1 prime power, and the same for n2. And the full transition probability is also proportional to the number of ways that we can pull out black balls versus white balls, which is just the binomial. So now we actually have a transition matrix describing the stochastic dynamics of, in this case, a neutral population. Right? If we want to incorporate the effect of selection, we have to bias the probability of pulling out a black ball versus the white ball. And that we can do simply by introducing it here, the following factor, delta x1, delta x2. Right? So this will incorporate selection, represent selection. Are you with me? I'm sure you are.
Even without
Yeah.
selection, where is the mutation rate in this transition matrix?
There isn't any. Because again, all we're doing is sort of representing the stochastic effect on so the propagation of genotypes in the population. Right?
So when you say that G1 can, okay, you're not saying that G1 can go to G2. It's merely that G1 can make
more copies of itself.
Okay, you're not
That's all.
You're not talking of transition
Not yet.
to G2.
Not yet. Not yet. But to some extent, that is simpler. We already have that as a stochastic process. So, but now we're trying to capture the aspect of stochasticity that we didn't have in this previous purely deterministic description. Okay, so this looks perhaps familiar or perhaps obscure. Yes, please. Well, I can't really go through sort of rigorous demonstration for you, but just intuitively, right? All that's happening, we're saying, okay, the probability is proportional to the frequency. But if it is more fit than average, we're going to increase that probability a little bit, right? Now, if there are only 2 genotypes in the game, then if this one is positive, this one must be negative, right? So this thing will automatically take care of one population being sort of doing better than expected purely by chance, and another doing worse than purely by chance, right? Okay. So for n much bigger than 1, you expect simplifications, right? So this is a binomial distribution. It's going to be very peaked on its mean. It will have some well-defined variance decreasing with n. So it will become rather sharply peaked on on the mean. And in that limit, it makes sense to talk about the frequencies. So we'll define the frequency of, let's say, the first genotype like that. And then, of course, the second genotype is going to have the frequency of 1 minus nu, right? Since they add up to n. So we don't need to keep track of both of them. It's enough to keep track of one frequency. And we can write down an equation for the probability of finding fraction nu of genotype 1. And that's going to be governed by a partial differential equation of a very familiar form. Nu 1 minus nu p of nu t. So in the absence of selection effects, that's basically what you would find. So you basically recognize this as a diffusion equation, diffusion in frequency, with diffusivity 1 over 2n, right? And there is this funky nu times 1 minus nu factor, and that thing is just telling us that frequency equal to 0 or 1 are special, right? Because if we lost all of the black balls, right, they're never coming back. Fine. So this basically just tells us that if we start with some sharp distribution, as time goes on, it's going to broaden. And eventually, what it's going to do is end up either all at 0 or all at 1. And there will be some probability associated with it, right? So the probability of an allele starting with some finite fraction and ending up at 0 is right, obviously gives us the probability of extinction, and right, but asymptotic states have to be either everything here or everything there, right? So if this allele did not become extinct, it has become fixed. And this equation will allow us to calculate this. And we can incorporate, right, basically rederive this thing in the presence of selection, and then we're going to pick up another term, which will look like this, with the x nu 1 minus nu of nu t. Right? So that incorporates the effect of selection. And this incorporates the effect of stochastic drift. And what you see here is that the larger the population, as long as we're thinking in terms of these macroscopic frequencies, so the larger the population, the less important is are these stochastic fluctuations compared to the effect of selection, right? it is also telling us that for the selection term to be visible, right, this delta x fitness differential has to be larger than 1 over n. Right? And if it is smaller, the dynamics will be swamped by diffusion. And it's rather unfortunate that the founding fathers have called the diffusion term drift. Right? If physics or engineers were doing it, right, we would have called this term drift. Alas, we're stuck with it. So, so that's one way of looking at it. But, oh, and I should say that this diffusion equation was derived by Kimura in the 50s. To my acute embarrassment, I don't quite remember whether this was 1950 or 52, but sometime in the 50s. So this is also known as Kimura's diffusion equation. So, any questions? So that's what you find in the textbooks, but actually it's turns out to be not the most useful way of making progress. So it's more useful to think about the stochastic process and genetic drift as a stochastic branching or birth-death process. So think of just a single individual. And there it goes on from generation to generation. And every so often, it makes 2 offspring. And these offspring sometimes make 2 offspring. But then, and let's call the rate of branching, let's call it birth rate, or branching rate, B. So obviously, if we just had this branching rate, right, this tree would be getting more and more branches. So in addition, we introduce some death rate. So as we go on, some of the branches are going to die, and some are going to live. And then it becomes the question of whether the death rate is equal to the birth rate, or which one is larger, right? Obviously, if the birth rate on average, well, birth rate is larger than the death rate, then the tree will still be branching. But even if the 2 are equal, right, something interesting will be happening, right? On average, the number will not be increasing, but the probability of extinction is going to be rather large, right? So there'll be a rather interesting situation where, with the large probability, there are no surviving offspring at all, but then there is also probability of having a very large tree, so that on average, the mean is working out right. So let's calculate that. So let's calculate the probability of finding exactly n live branches in this tree after time t plus delta t. So we're thinking of this as a continuous or Poisson random process in time. So that probability is equal to what it was before at the earlier time, provided that nothing happened. And I guess I should have said here that these rates are, right, they're rates, so the probabilities in the time interval dt are given by this rate times dt. So there is some probability that there is no branching event in interval dt, and none of the existing branches have done it. So n times pn, no, I don't need pn anymore. I already got that. And we want also to so correct for the probability that any of these existing branches die. On the other hand, if we do have a birth event, right, the number of branches is going to increase by 1. So probability of n minus 1 is going to contribute to probability of having n. And similarly, the death event is going to take any of n plus 1 individuals and turn that into n. That's what we got. And time scale. So let's choose the time scale so that the death rate is 1. And the birth rate, we're interested in a situation where it pretty much compensates the death rate, except maybe the fitter individuals are branching a little bit faster, or less fit individuals are branching a little bit less fast, right? Yes, please. I'm sorry? Right. So we're trying to calculate the probability of finding at time not a terribly good picture. But we're trying to define the probability of finding exactly n branches at time delta t and relate it to the probability of having n branches in the previous time slice, right? And the first term just says that if nothing happens during this short interval, then this probability, right, this probability is equal to whatever it was in the slice before, right? And this bracket just calculates the probability that nothing happened. There was neither birth among the n members of the population, nor death, right? Nobody died, nobody branched, right? This term accounts for the probability that any one of the n minus 1 members of the population branched in that time interval, right? And that changed the number from n minus 1 to n, right? And similarly, exactly the same thing for n plus 2 if it was the death in the family. Okay. So let me just do a bit of arithmetic quickly. I just turned this sort of discrete equation into a differential equation. And I missed a factor of n here. And now we have to solve it. So there is a very useful trick in probability theory. It's called generating functions. Generating function trick. So what we're going to do is define, just by fiat, the following function. We'll introduce a variable z and define a function like that. So this is basically a relative over Laplace transform. What we're doing is a little linear transformation of our system of equations to manipulate it into an isoform. And as a homework, you just take this equation, multiply by z to the n, and sum on n. And work out the consequences and convince yourself that what you get is the following. Now you're going to have a partial differential equation of this form. Oops. You write down an equation like that, and at least when I do it, the probability of me making a mistake is very high. So I immediately need to check something. An easy thing to check is the normalization. If we substitute z equal to 1 here, right, we don't have to worry about that. We're just summing the probability over the independent variable. So g of z of t when z is equal to 1 is always equal to 1. So it is time independent. So it better be that our right-hand side vanishes when z is equal to 1. And thank you very much, it does. So at least I passed the first test. So actually, how many of you know how to solve equations like this, right? First order. Yes. Linear equation. Come on, you're just not confessing to it. I'm sure you know. But the way you solve this is by method of characteristics. Which is basically a nifty little trick that always works for equations like this. And essentially a statement that if we look in a plane here, z of t, there is a curve somewhere in this plane. We call it zeta of t, such that our function of interest along this curve is equal to a constant. So then the whole story boils down to finding what is this curve, right? Of course, we're interested in the curve that ends at our point of interest z here, right? And let's see how I'm doing on time. You know what? Solving this is going to be maybe homework. Or maybe I'll tell you just one more thing. So again, at home, you can convince yourself that this curve itself obeys a simple equation, which is basically dt g of zeta d zeta of g. Which is exactly this factor here. So that's what you want to solve. And since I'm running out of time, let me just try to give you a magic solution. Can you see this? It actually has a rather simple form, right? So, well, I guess I'm skipping a step. I'm not telling you exactly what this curve is, but just giving you the answer for the generating function. And it has very simple form. It is basically a linear rational function with two coefficients that are dependent on time and calculated here completely explicitly. So let's just not worry about exactly where this came from. You'll derive it at home if you want. But let's talk about what it means. Well, disappointed. I really wanted to actually do a calculation on the blackboard, but I'm running out of time, and I'm skipping a bit of arithmetic. But maybe you're happy with that. So let's try to understand what the hell is going on. Yep. Okay. Except on the screen, you can't see the most important part. So we were interested in this extinction probability. Remember, I just erased this expression that we had, P of n of t z to the n. Notice what happens if you substitute z equal to 0 here. Generating function evaluated at 0 gives us exactly the probability of no branches surviving. This is extinction probability. Okay? Now we go to our little expression. And, okay, we have 1 minus z here. We substitute z equal to 0, z equal to 0. This whole thing becomes 1. So our a, the function of t, here factor that we have calculated it explicitly. Now I can point on the point on the whiteboard here, right? This factor a of t is exactly the extinction probability that we're after. So let's just think through. That's what we wanted, right? Let's just think through what it gives us. So first, let's consider the neutral case, delta x going to 0. So we have to be a little bit careful. The numerator here is going to go to 0. But the denominator, right, delta x is getting small, so the exponential here is getting close to 1. 1 minus 1 is 0, and this is supposed to be 0. So the numerator is 0, denominator is 0. At school, we'll learn to be very careful in situations like this. And you work it out and convince yourself that p of t then is exactly equal to 1 minus a, which comes out to be 1 over 1 plus t. So at time equal to 0, extinction probability is equal to 0, right? We have our branch right here, right? But at long times, this term is going to 0, and the extinction probability is going to 1. Okay, and now we can work it out also, for large times. For the other cases, for example, when our allele has a positive differential fitness, you will find the extinction probability behaving like this. So now extinction probability doesn't go to 1 in the limit of, well, I may as well say very long times, right? 1 minus extinction probability is survival probability. So the survival probability is basically proportional to that fitness advantage. On the other hand, if delta x is 0, then our magic expression will give us 1 minus delta x e to the minus delta xt. I'm putting in a modulus to turn a negative number into a positive number. And now the second term contains this dying exponential. So again, the allele is getting extinct with probability 1. And it is approaching extinction much more rapidly than it does in the neutral case. Right? So we're actually managing to learn a great deal from the simple calculation. And we were just 10 minutes away from doing the whole bloody thing on the whiteboard. But this is close. And we're not done yet, because we can calculate more. We can actually calculate this probability distribution completely. All we need to do is take our relatively simple expression and do a Taylor series expansion in Z. Basically turn this expression, pointing with my finger, total lack of class, into a power series like this. And then term by term, we'll identify basically coefficients in the Taylor series will give us these probabilities. And what you'll find, let me just do it again first for the neutral case. You will find a simple expression. You'll find the probability of still having n copies surviving in the population is exponential in n. but the prefactor of that exponential is getting smaller with time. So at long times, the probability of having a large deviation, having large n here, is higher than it is at short times. On the other hand, this whole thing, right, this is conditional on survival. And this prefactor is getting smaller with time, right? And it's the same prefactor which governs the survival of any offspring at all, right? Non-extinction. So the picture is basically following. As a function of time, if we were to follow individual branching processes in time, we would have something like this. It would start, let's say, at 1. Yeah, that picture. Would start at 1, and then with high probability go to 0. But if it doesn't go to 0, then it would actually deviate a lot. And so on and so forth. So basically, any allele that has not become extinct by time t has a good chance of reaching a copy number that is proportional to t. So what happened to our fixed population size? We actually threw it out when we started doing this branching process. And that's sort of a beauty and the simplicity of it. Very often in actually trying to calculate the rate of adaptation, right, we'll be interested in the fate of new mutations. Right? These new mutations come at copy number 1. Number of copies equal to 1. So in a very large population, it's not terribly useful to think about the frequency. The frequency is 1 over population size, some very small number. It's better just to count them, right? And on the other hand, we'll be interested in so most of the effect of, most of the extinction is going to happen right away, before the number of copies rises to a significant fraction of the population size, because then that already is going to be a large number of copies. It's not likely that they'll be extinct. So as long as we're thinking about these relatively small numbers, it's much better to work with the branching process. And then the overall constraint on the population size, the total population size being fixed, comes in later. But very crudely, we can even estimate the probability of fixation. So we were just going to say that somewhere out here, there is the total population size. So street fighting mathematics, sloppy, but catching the main point. Now the probability of n, of a little n, number of copies, to reach this full population size, to take over the whole population, depends on, the characteristic size, the characteristic number of copies increases linearly with time. So in order to get to capital N, we'll typically have to wait for the time of order n. And at the same time, the probability of surviving that long is going to be 1 over n, right? So from this very crude argument, we just learned that fixation probability is of order 1 over n, and fixation time in a neutral population is of order n. Right? And we can't quite get a hold of a prefactors here from this crude argument. But one can actually calculate that in this full Kimura diffusion equation, and then the correct answer is going to be exactly that. But we're clearly on the right track. So, I think I'm just going to stop right here for now, because my goal was to walk you through a little calculation, and I think I have, even though I skipped a bit. And just to remind you why we were doing this was calculating this survival probability or extinction probability is going to be the main ingredient in understanding sort of the effect of stochasticity on the fate of new mutations. Right? And I gave you a flavor of how to do it, and Rob graciously agreed to share a bit of his time with me today. So we're going to come back later, and I'll again streamline and tell you how you can use these ideas to calculate rate of adaptation.